So after we've cut pieces of DNA into the sizes that we're interested in working with, we may need to separate those fragments and figure out what size they are or what genes they contain and what we're going to do with them next. Okay, so usually the first step is to separate the fragments of different sizes using a technique called gel electrophoresis. Okay, this not only separates the fragments into different sizes, it also involves a special radioactive dye that allows the DNA to be visible. Okay, so what we do when we have a sample containing multiple pieces of DNA is we set up what's called a gel that has multiple lanes, and it has a well at the top for putting the DNA in. Okay, so this gel is made of a substance called agarose. So it's kind of a flexible, sticky substance that has a lot of pores inside it. Okay, and we'll talk about why that's important in a moment. Okay, it's going to be attached to some electrodes. There's a negative end and a positive end. And there's a hole or an indentation in the gel at the loading end. Okay, and you always want to load the DNA at the negative end. Okay, do you remember from our study of DNA whether DNA is positively or negatively charged? Okay, hopefully you remembered that DNA is negatively charged. Okay, so when we turn on the current, which direction is DNA that's placed here going to move? Okay, it's going to move towards the positive end. Okay, and if we load it at the wrong end, or if we hook up the electrodes backwards, the DNA is going to go this direction and it's going to slide right off the gel and we won't be able to use it. Okay, so once we put our DNA sample in the indentation, known as a well, it's going to move along this direction, and the area below the well is known as a lane. Okay, we can have multiple lanes on our gel, so we can put different samples in each well and compare how large the fragments are that each different sample contains. Okay, after we turn on our current and we have the DNA moving for a while towards the positive end, we're going to have the fragments of different sizes moving at different rates. Okay, they're all going to be moving in the same direction since all DNA is negatively charged, but the smaller fragments are going to be able to move faster than the larger fragments. Okay, and sometimes we have another well that contains fragments of known sizes that we can use to compare to our unknown columns. Okay, now I want to note that the rate at which these fragments move is not always directly proportional to their size. A smaller fragment is always going to move faster than a larger fragment, but a fragment half the size of a larger one may not move twice as fast. Okay, you tend to get a little more bunching up of these larger fragments. So keep that in mind when you're reading the results of a gel and trying to interpret what it means. After the current has been running for a sufficient amount of time in order to get the bands of DNA to separate adequately, okay, and that amount of time depends upon how big your fragments are, how strong your current is, and a lot of other things. Okay, so you need to check your gel after a while, see if most of your bands are still up here relatively close to the top. You want to run it a bit longer. If many of the smaller fragments have made it down here, you want to hurry up and stop it before they slide off of the gel. Okay. The DNA needs to be dyed with certain markers, okay, either chemicals or fluorescent substances are added to the DNA fragments so they'll show up properly. And a special lighting system is put underneath your gel so that it can be viewed accurately. Okay, so these thicker bands contain a lot of DNA, and a thinner band would contain less DNA. And the fact that these are especially bright means that there's a lot of DNA there too as well. Okay, so it's important that you're able to look at a gel and tell its relative size by comparing it to the position on the gel of other fragments. Okay, so a quick review here. Let's say that you have 3 DNA fragments in your sample. One is 500 base pairs, one is 1,000 base pairs, and another is 2,000 base pairs. You're using a gel electrophoresis, to separate the sides of the fragments. Which one is going to move the farthest in a specific amount of time? Okay, hopefully you said the 500 base pair fragment. Okay, the shorter the fragment, the faster it's going to move. After we've separated our DNA fragments, we need to know which piece contains the gene that we're interested in. Okay, let's say that we're studying a bacterial genome. We've used restriction enzymes to cut up DNA into 7 or 8 pieces, and we've separated them with gel electrophoresis. Okay, we're looking for a particular gene. Okay, we're going to use something called a probe to locate where that gene is. It's basically going to stick to some DNA fragments and not to others. So probes use the concept of complementary base pairing. Okay, if it's a DNA probe, it'll pair with the DNA segment that it's complementary to. Okay, so we'll need to make sure that that fragmented piece of DNA has been heated up and the 2 strands have been allowed to separate. If that probe is RNA, okay, we want to make sure that that RNA sequence is also complementary to our piece of DNA that we're looking at. Okay, so the first type of probe to be developed was developed by a person whose last name was Southern. Okay, so that was called the Southern blot. It was using a DNA probe and the principles of complementary base pairing to detect DNA of a certain sequence. Okay, and that sequence has to match up pretty well with most of the base pairs being the same and in the same order, but it is robust to allow a few differences. Okay, so let's say that we're talking about a particular gene and it may have a point mutation or 2. Okay, it might have a base pair substitution, but it's several hundred base pairs long. Okay, the correct probe should still be able to find it. After Southern developed this technique, and it was named after him, the Southern blot, other related techniques were also developed. Okay, the Northern blot was made to detect RNA, but there was no one named Northern. Okay, it was just a cute name to show that it's a contrast to Southern. And then later, Western blotting was a mechanism to detect the presence of certain types of protein. Now, why do you think you'd want to use these different techniques in different situations? If you're looking for DNA and for RNA, you should be able to find the same gene, shouldn't you? Because every DNA sequence has a complementary RNA sequence, okay? And during transcription, RNA is made from DNA. So it's true, you can get a lot of the same information from a Southern blot and from a Northern blot, But why might you sometimes want to use one or the other? Okay, if you're working with a bacterial genome and you have only a single cell, there might be not so much of an issue. But you still have certain different environmental conditions that cause transcription to occur in some cases and not in others. So suppose you do a southern blot, you detect that a particular gene you're interested in is there, but you also want to test the cell under different conditions. And sometimes you find the complementary RNA, okay, the messenger RNA that's transcribed, and sometimes you don't. Okay, then you would know that those particular conditions in the cell are responsible for regulating transcription. The same issue is around with protein. Okay, you might find a situation in which gene expression is not regulated at the level of transcription. Okay, transcription occurs in more than one situation, but translation may be blocked by one of those post-transcriptional measures. So you can see why it's useful, depending upon your research question, to have access to these different types of techniques. Okay, after we've separated DNA of different fragment sizes, or maybe even before we cut it with a restriction enzyme to begin with, we sometimes need to amplify or increase the amount of DNA that's present. Okay, some molecular techniques require large amounts of DNA and not just one particular copy. So fortunately, scientists have developed a very effective mechanism for copying DNA very quickly and in very large amounts. This is known as the polymerase chain reaction. Okay, polymerase refers to the enzyme that copies DNA polymerase. Okay, so developing this technique required discovering a particular type of DNA polymerase. Okay, it had to be stable at very high temperatures. Okay, remember from your intro bio course how high temperatures often denature enzymes. Okay, and each enzyme has a particular temperature through which it's stable. Let's take a closer look now at how this occurs. Okay, the reason that we needed such a special type of DNA polymerase that was stable at such high temperatures was that in order for DNA to be copied, the two strands had to be separated. Okay? This requires the breaking of the hydrogen bonds that hold the bases on each strand together. High temperatures will cause these bonds to break down, but will allow the stronger bonds in the backbone of DNA to stay intact. After heating up the DNA, it's quickly cooled to a lower temperature, and primers have to be added. Okay, remember that replication of DNA requires the addition of an RNA primer. The solution is then heated again slowly, and the DNA polymerase synthesizes the complementary strand, which creates two new double-stranded DNA molecules. Okay, this cycle can be repeated over and over again. Every time a cycle is completed, the amount of DNA doubles. So it's possible to get millions of DNA molecules, exact copies of the ones you started with, within only 20 or so cycles. And it's possible to get 1 billion pieces of DNA or 1 billion copies in 30 cycles. Okay? This was a revolutionary new technique. Okay? It was responsible for allowing a lot of people to be released from prison because they were DNA evidence was available for information about their trial, but we didn't have the technology to amplify the DNA enough to sequence it and figure out what it actually meant. So now we can take like a tiny drop of blood or the root of one hair and make enough DNA in order to do a lot with it. Often we want to clone genes in other ways, other than a PCR. We can amplify or copy a specific piece of DNA using the machinery in a bacterial cell. This bacterial cell is going to replicate over and over again and copy the target DNA molecule along with its own DNA. So often we use what are called plasmid vectors. Okay, think about the bacterial genome. Remember that a bacteria has a large circular DNA chromosome, and it also often has several smaller plasmids. Okay, remember those are smaller pieces of DNA that are also found in the cytoplasm. And scientists can insert foreign DNA, or the DNA that they're studying and want to copy, into the bacterial plasmids. Okay, so the restriction enzymes that we talked about before are used to cut the DNA. The foreign DNA is introduced, and sometimes we need special fragments that are complementary to those sticky ends to help us get that DNA in. The bacteria that have taken up the foreign DNA are known as the transformed cells. When we try this, it's not going to work with 100% success rate. Okay, we'll try this in a culture of bacteria. Some of the cells will properly take up the foreign plasmid, and some of them won't. So we then need to screen the cells for these recombinant plasmids in which the taking up of foreign DNA has actually worked. Sometimes we can use a probe to figure this out, and there are other markers or indicators that we can use to confirm whether or not a particular cell has been transformed and indeed contains the particular piece of DNA that we're interested in. Okay, besides bacterial plasmids, we also use other types of gene vectors. The plasmids are generally small enough that they can only use, or they can only contain, a relatively small piece of DNA. So this list here shows, usually in increasing order, vectors that are allowed to carry bigger pieces of DNA. One example would be a cosmid. Cosmid is a virus that has had all of its own DNA removed. Okay, so there's space for the foreign DNA, and that allows for a little bit more space than most plasmids do. We can also create what are known as bacterial artificial chromosomes. So these are a similar concept to the plasmids. Bacteria often conjugate or exchange genetic material with other bacteria, and they have a plasmid involved in mating or in exchange with these other bacteria that tend to be a little bit bigger. So scientists have modified these larger plasmids and allowed them to be able to contain larger pieces of DNA that they're interested in. We can contain even more DNA if we're working with yeast. Okay, so think for a moment. How is a yeast different from a bacterium? Okay, hopefully you said that yeasts are eukaryotes while bacteria are prokaryotes. And eukaryotic genomes are much larger than bacterial genomes. Remember that yeasts and many other eukaryotes can be unicellular. Okay, so a yeast artificial chromosome is going to be able to contain even more foreign DNA. And the TI plasmid is a specific type of plasmid that's known to be very large and can also carry a lot of DNA. Let's take a look at how this works. Okay, we're going to graphically go through what happens when we insert a piece of DNA into a plasmid. But before we do that, we've got to make sure that we've selected an appropriate plasmid. First of all, the plasmid has to have a site for an origin of replication. Okay? It has to be a plasmid that can be copied. Inserting it once really doesn't help. We need lots of DNA, so we need to use that bacterium or that organism to copy the DNA that we want over and over again. So we got to make sure that there is an origin of replication. Next, we've got to make sure that there's a selectable marker. Okay? This is a particular trait that allows the colonies to be identified. This might be something that makes one bacterium look different than another, or makes it react to an enzyme differently than another. Okay? When we try and do this, it's not going to work 100% of the time, and we have to be able to choose the plasmids that are in cells that have taken up the gene of interest. So their natural or wild type gene is going to be disrupted. Third, we need to have a cleavage site for a restriction enzyme that's unique. Okay? So whichever restriction enzyme we choose has to cut at only one place on that particular plasmid. So if we choose a restriction enzyme with more than one location for cutting the DNA, we either need to choose a different plasmid or a different restriction enzyme. We don't want this plasmid cut into many pieces. We just want one opening to insert our one gene that we're trying to copy. Okay, let's take a look at how this works, given the fact that we've already identified an appropriate vector for copying our DNA. Okay, so we've got our circular plasmid here. We're going to make a cut with one enzyme. Okay, remember there's two sites because it makes these jagged end cuts. And then we're going to insert our DNA into the medium, and hopefully in some of the cells, the foreign DNA with the complementary ends, okay, that we've engineered to fit with that particular complementary sequence, that'll be taken up by some of the plasmids. A few of them won't take it up, okay? They might seal themselves back up before the new DNA gets in. But that's fine. We've got a culture of many bacteria. If it works even a small percentage of the time, it's going to be successful. After that piece is inserted, DNA ligase closes the gaps. Remember, these nucleotides have to be joined, and DNA ligase is very good at sealing single-stranded breaks. Okay, so now we have the plasmid that we're interested in. Now we've got to figure out which plasmids or which bacterial cells have the plasmids that have taken up the DNA that we're interested in. So here's a common way of doing this in E. coli cells. Okay, if you think back to our chapter on gene regulation, and you remember that one of the operons we studied was the lac operon system. Remember the LAC-Z gene for beta-galactosidase? That was one of the structural genes controlled by the lac operon. Okay, if we have a plasmid in E. coli that contains that gene is known to be able to produce a product with a blue color when it's mixed with a certain chemical called X-gal. So if we have an intact beta-galactosidase gene in a plasmid, we put in the X-gal, we expect blue colonies to form. If the beta-galactosidase gene has been disrupted by our new gene that we've inserted into the plasmid, we don't see any turning blue. So those bacterial cells will remain white. We then plate our bacteria. Okay, remember when we've got bacteria growing in solution, we can have thousands or even millions of cells in a very short period of time. We spread them out on a plate, using dilute medium, incubate the plate, let them grow for a while, and each of these spots produces a colony with many cells. We then know that the white colonies have taken up the foreign DNA, and the blue colonies have an intact gene for beta-galactosidase. So we can then remove one or more of these white colonies and grow them up and make lots and lots of copies of our target DNA. Another way that we can use an identifiable marker to select for the colonies that have our DNA of interest taken up is to look for antibiotic resistance. Certain genes allow bacteria to resist being killed by antibiotics. And remember when we talked about gene regulation, okay? We talked about sequences, including promoters and operons, as well as transcription initiation sequences, okay, that allow a gene to be turned on or turned off in certain situations. Okay, these situations could be the presence or absence of a particular molecule that binds with a substrate and turns on or off the gene. If, in inserting our DNA gene of interest, if we disrupt a site that allows a gene to be turned off during certain conditions, we can create those conditions, and in cells in which no foreign DNA was taken up, transcription will be repressed, and the gene that confers resistance to the antibiotic will not be transcribed. In that case, we can expose the colonies to the antibiotic. All of those cells will die, okay, because the antibiotic will kill them. In the genes where we've disrupted the operator, or we've disrupted something that will turn the gene off, that gene cannot be turned off and transcription will be occurring. This antibiotic resistance gene will be produced along with the DNA that we're interested in, and we can simply collect all of the colonies that are still alive after exposure to the antibiotic, and we know that those cells have taken up the DNA of interest.